this one too for the stream. Wear both.
Both? Okay.
Yeah.
Yeah, I'm just standing here, so I'm not running around.
Keep this one. That's the crappy.
Clasp on it.
Yeah.
Sorry about my collar.
No, it's okay. Just trying to figure out the best way to do this. There we go. That's good. Is that,
They're both all right?
yeah, that's the union one, so I'm not in control of that one at all. So you'll turn that one on. This is just for the stream.
All right, do I have to flip this one on as well?
No, it's already on for you.
Okay, thank you. I'll talk about that. All right, if you can go ahead and get seated, get ready. It is 10:30, so we're going to go ahead and start. Feel free to move close to the front if you need to be able to see or to hear me, and also so I can hear you if you ask questions at any point. All right, so this is the study union final exam review for genetics. I'd like to ask that if you are not here for genetics, that is, you're not in a course with either Dr. Bayer or Dr. Sotero, that you please step out for the privacy of your fellow students who are in this course. If you're staying, I'm going to assume that you are here for the actual review. I'd like to make a note that this session is currently being streamed via YouTube, and that it will be recorded, and you can visit the SARC YouTube channel afterwards, and this whole session will be recorded and uploaded there. You've probably noticed if you visited the study union site that what was up was not any sort of problem set or outline like I've done in the past. It was a set of topics that you should have reviewed prior to coming here. That's how you get the most help out of it. I decided since your test is 24 hours away. What I'll be going over today is a problem set of 40 questions. You probably noticed that this isn't online yet. And I'd like to say that after we go through it and after we leave the session, I'm going to be uploading it onto my own genetics site. If you've been in Bayer's class, you've probably been on the site before, but I'll go ahead and show it. This is the site with the topic guide, and also if you have any other classes that are holding a study union, this is where all your materials will be. And then afterwards, I will upload the practice set and the key onto this site here. I'll show this again at the end so you can take it down. All right, I did mention that the two classes in here are a mix of Dr. Bayer and Dr. Sotero. Broadly, the majority of the material will be the same, but you may notice maybe one or two questions if you are in Dr. Sotero's lecture that may be, oh, I'm not familiar with that, and that's just differences in material that professors cover. Do not be alarmed if you see something unfamiliar and you're in Dr. Sotero's class. Just go with what you've learned in lecture. And on the other hand, if you think I missed something, keep in mind that maybe Dr. Bayer didn't cover it, since it is geared toward her lecture. If you notice any errors as we're going along, feel free to let me know. Either raise your hand and point them out, or afterwards. What I'm going to do is I'm going to give you guys like a minute or so to work on the problem. And during that time, you can feel free to talk quietly with your neighbor. I encourage group work. Figure it out, and then we'll take a minute and discuss the answer. And then I'll try and leave a bit of time at the end. We go until 12:30. I'll try and leave some time at the end so that if you have any questions, just keep in mind there's a lot of you in a limited amount of time. And with that, Oh, I would like you to know that I don't see either the final exams. This is sort of like my best guess at what you might see based on just being in your class all year. And again, if there are any errors, please let me know. All right, so question 1. Which of the following statements is correct? So go ahead and take a minute and look at the statements, and we'll convene again in a minute. Yes? Yeah. There we go. Is that more visible? Cool. For those of you who are either just joining us in the stream or just filtering in, since it's still in the first 5 minutes, we're just working on this problem right now. We're going to go through about 40 questions, so feel free to come on in and start working on this. All right, so this problem was just one to give you that's probably for most of you a review from Bio 1, just a basic difference between prokaryotic cells and eukaryotic cells. And it would be that prokaryotic cells contain a singular circular chromosome within their nucleoid region. So eukaryotic cells have a nucleus, prokaryotic cells have a nucleoid region. Eukaryotic cells have linear chromosomes, prokaryotic cells had circular chromosomes. So this was just a simple difference between cells. Any questions? Okay. Between each question, I'll pause for questions. Okay, this was also maybe going back towards Bio 1 or towards the beginning. So the definition of an allele. What did you guys get? A, that was a resounding A. So an allele is the form of a gene found at a given locus. Does anyone remember what the locus is? So the location along the chromosome. It's even got the word location in its name. So knowing what an allele is will be important when we get to Mendelian and heredity. Okay, we're going to go ahead and move on to the cell cycle. All right. So what did you guys find for the answer? So C. So we have what occurs during the gap or G0 phase. So it's just going back to your cell cycle during interphase. So when you look at a diagram of the cell cycle, it's circular. And up here you would have your mitosis. And somewhere along the way you have a gap 0 phase. Oh. Sorry about that. My mic cut out. Can you still hear me? Okay. I'll try and be as loud as possible right now. So just let me know if you need me to be a little louder. So the gap 0 phase is when a cell will exit the cell cycle. And think about, for questions on the test, think about what does that mean for it if a cell exits that cycle. Not only what is it doing during that time, but what doesn't it do? So now we're going to go ahead and move on to a question on meiosis, and I'll give you a minute to work on this one. All right, so this one just required you to choose the correct statement, and I was asking about the types of division and the division outcomes between meiosis 1 and meiosis 2. This one required you to read a little bit more, look into the details. So what answer did you guys come up with? So I heard C, some B, B. Okay, so this introduces terminology maybe you guys haven't looked at since we were in this section. The idea of meiosis 1 being reductive, and then meiosis 2 is equational. So can anyone tell me what reductive refers to? Go ahead.
I thought meiosis 1 creates two diploid cells, and then it goes into meiosis 2 being 4 haploid cells.
So if it produces diploid cells, would it be reductive or equational? So I hear equational. So equational, we would see if it's diploid, it would stay diploid. So in here, we see it goes from 2 haploid cells to 4 haploid cells. So it divides, which will increase the number of cells we have. But because it's equational, that haploid number, that N number, won't change. So we look at reductive. So it was a good thing you brought up that it starts diploid and it goes to haploid. So this word, go over here. Reductive, a reductive division, in terms of meiosis, we're going from a 2N count, or a diploid, to a haploid state. So just some vocab that will be helpful. Anyone still confused on this? Yep, you're going to have to speak up. It doesn't change the N. So, thank you. Yeah, so you look at that word equate. So it doesn't change the N number. Of course, because it's a division, we will always increase our number of cells because a cell is dividing. But the equational slash reductive will always be what's telling you about the diploid haploid number. All right, so this question's just asking about chiasmata. Another important sort of vocab word for you. Okay, so what did you guys find for this one? So I heard D. So areas where crossing over occur. So can anyone tell me what the kinetochore is when we're talking about that in answers A and B? So I can't hear you guys, so just one person raised their hand. Go ahead. So the kinetochore is where spindle fibers attach. And those spindle fibers will help us when we're pulling apart these chromosomes or these pairs and migrating them across the cell. So here's a question. So is there significant crossing over between sister chromatids? So no. Why? Because they're identical. So if you swap areas of the chromatids and they're absolutely identical, we wouldn't even be able to tell if they crossed over at all. So what crosses over? So homologous pairs. So now we're going to go on to a little bit about Mendel. I know some people came in during that last question, and if anyone's tuning in online, we're just going through a problem set. It'll be about 40 questions, so we're just about a chunk of a way into it. Feel free to talk to the person next to you. It would help if you've started reviewing the material. Also, unfortunately, my mic has died, so if you cannot hear me, especially some of the folks in the back, please let me know and I will try and speak up a little bit. Or if you want to move forward to hear me, that's just fine. All right, so this was just asking you to think about just the broad conclusions that Mendel came up with and what some of his laws were saying. So in a set of heterozygote parents, which of Mendel's conclusions lead us to predict that about half of the gametes will inherit a dominant allele? What did you guys find? So I heard D. So what does the law of segregation state? So keep in mind when you're answering a question like this, in the back of your head when you're looking through all these laws, you should be able to think to yourself, okay, well, what does that law state? Because otherwise it's going to be kind of hard to just try and figure it out, especially because his two main laws, independent assortment and segregation, they sound really, really similar. So can anyone from their notes give me a definition of Mendel's law of segregation? Yes, you in the back. So, alleles, let's see this all right. And who can differentiate that with the law of independent assortment, or give me a definition of that law? So can anyone confirm that with their notes? I heard yeah. Yeah, someone's pulling that up. So random alignment of chromosomes. So I'm going to abbreviate that, just chromes. So then the answer, you guys agree, is D? Can I explain the difference between the two? Yeah. Okay, so I'll pull this back down. All right, so here we were talking about we have two of Mendel's laws, and one had to do with the alignment of chromosomes and the idea of how they line up along the metaphase plate, and one had to do with the movement of alleles and how they segregate. So keep these laws in mind when you get to crossing over. So, and go ahead and review these slides on your own as well. Like I said, does anyone still, you disagree or? Okay. So go ahead, and if you're still confused, you can review it in the slides from your lecture or in your text. Now we're going to move just to an example of one of the different sex determination systems. Okay, so what did you guys find for this one? So, C. Why C? So, okay. So, who remembers the three main types of determination, the main types of determination that we were looking at in class? So we talked about genic. What's another one? Environmental and chromosomal. So which one of those three do you think deals with the sex chromosomes like our XY, ZZ, ZX systems? Chromosomal. And then we talked about environmental. So sounds like it would use the effects of the environment. So if we know that chromosomal deals with the sex chromosome systems, then what do we think genic would lead to then? So I heard gene sequences, and now your sex chromosomes do have genes on them, but there's a differentiation between whether you're looking at specifically the sex chromosomes or the gene sequences. So under genic, we are still looking at the genes rather than environmental conditions, but instead we're looking at other genes. So it would be D. Because as far as the actual chromosomes, we'd be looking at in the sex chromosomal base determination. Versus genic, we're still looking at genic possibilities, but not necessarily, oh, one sex has this chromosome and the other sex has this combination. Any questions on that? So make sure to keep that straight in your head. And with this kind of thing, when you're dealing with multiple systems, remember what the others entail, because there is a more specific name for when we're looking at sex chromosomes. I want to return to the last question real quick. So I think it got a little backwards with the laws of segregation and independent assortment. Got me thinking when I mentioned linkage. So this is on page 51 of your textbook of the 5th edition. And here's a summary and comparison of these two different principles. So here it says that the law of segregation has to do when the alleles separate and they separate in equal proportions. So the question I asked you was that half would get the dominant allele and therefore half would get the recessive. Whereas independent assortment has to do with different loci. So if genes are not assorting independently, what do we refer to them as? So linked genes. So this has to do with the alleles along different genes and whether or not they move together. Where segregation more has to do with the alleles separating in equal proportions. So over here then, what would the answer be to number 6? It would still be then what? The law of segregation still. So I just wanted to clarify that. So, and this has to do with, another way to put this is not linkage. So I just want to clarify that. Okay. My apologies. Okay, so which answer did you guys come up with? So B, Turner syndrome. So what stood out to you that made you think it was Turner syndrome? So the webbed neck. So that's a big thing that's really sort of unique to Turner syndrome, is that webbed neck appearance. And also a thing to keep in mind when you're looking at these different conditions, make sure to check the sex. If you're getting like a description, to see, to check the phenotypes. And then another thing would be to look for fertility and also cognitive development and intellect. So keep those in mind. If you're having trouble keeping them straight, you can always make like a table and think, okay, is there anything really interesting about it, like the webbed neck? What's the sex? What is the intellectual development like? And then what about the fertility? Oh, quick question. What would happen if an individual had 2 Y chromosomes? So, death. So the lack of even 1 X chromosome leads to death. You can't have an individual who will survive to anywhere near term without an X chromosome because it's huge. It's got all this information on it. Okay. Now we're going to look at some of the extensions on Mendelian principles. Okay, so this question was asking about codominance. So what answer did you guys come up with? So E. So in each of these you had to figure out, okay, what does the heterozygote look like and what does a homozygote look like? What's the definition of a heterozygote? So 1 dominant, 1 recessive allele, so it has 2 different alleles. What about homozygous? 2 of the same, and you can think hetero is 1 of one and 1 of another. Homo is same. So here, heterozygotes will express both of the phenotypes, while homozygotes express only one. What's an example, sort of like a really big example in science of codominance? So tortoiseshell cats, is that codominance? Or is that more related to something else? So X inactivation. What's like another sort of like really big one in science? So what? So we like to talk about flower coloration. So what would a heterozygote, like flower, if you had one of the traits was red and one was white, what would a codominant heterozygote look like? So patches of red, patches of white. So it would display both. What if it was pink? Incomplete dominance. So do you guys remember blood typing? what kind of dominant system is that? So yeah, if you look at AB blood typing, if you inherit an A allele and a B allele, you make both antigens. So that's one way that we look at a codominant system. That one's a little more complicated if you think about the Bombay typing, but that's sort of like the really big example in science, especially if some of you have had labs. Might help you out a bit. Okay, now we have a question on pedigree analysis and forms of inheritance. Okay, so in this question, we had a pedigree analysis who is examining the pattern of a trait within a family. He finds that this trait appears equally among both males and females, that at least one parent of those with the trait will also have it, and that the trait doesn't skip generations, and that those lacking the trait don't pass it on to their offspring. So what sort of trait most likely are we looking at in this family? So A, autosomal dominant. So let's break this down into two parts. So what made you think that it was autosomal versus X or Y linked? So equal among the sexes. So there was that distinction that it doesn't occur more in females or exclusively in males. What would the sex distribution look like in Y linked? So it would be all in males. And what led you to believe it was dominant? So doesn't skip, and then we can look at how it's inherited. So if someone has the trait, or they don't have the trait, they're not going to be able to pass it on. And then if someone has it, at least one of their parents had to show it. So always break these down in dominant and recessive. What's another sort of common trait that I didn't mention in the problem about autosomal dominant traits? It's less of a pattern and it's more of like a common thing. So is it more common or less common? So, okay, so more common. So which of these do you think then would be lethal, more lethal than the others? Which one? So if autosomal dominant is sort of like the more common one, what about the more lethal one? So the recessive. So a lot of times, look at the patterns. The recessive tends to be, we see higher rates of lethality. And of course, we need 2 copies of it. a quick jump back to mitosis and meiosis real quick. So when does crossing over occur? So C, the prophase of meiosis 1. So this again has 3 parts to it. Break it down. So it's during prophase. So right away that would have told you which one it was if you just remembered that it was during prophase. And I had one answer that was mitosis and the rest were meiosis. So knowing that it was in meiosis, how do we know right off the bat that it doesn't occur in mitosis? So in mitosis, thank you, it produces 2 identical cells. So barring mutation, we don't see a lot of variation during mitosis. And then remember whether it occurs during 1 or 2. So again, just general tips when you're taking tests, especially when you get sort of later on. Break down the answer options, because again, this answer had 3 parts. And I could have given you very similar answer choices. So brought it up before, and now here it is, a question on linkage. Okay, so for this question, we were talking about a complete linkage, is we can have it be completely linked or incompletely linked, and with no crossing over. So if we crossed a heterozygote with a homozygote with complete linkage and no crossing over, what would our progeny look like? So E. So they would be non-recombinant. So what does it mean when it's non-recombinant? So it looks like its parents. So we don't see a recombination of alleles. So think about that. So we're not generating new combinations of alleles. We're not generating new phenotypes. So then it would make sense, the second part, of a parental phenotype ratio of 1 to 1. So half the offspring would look like one parent, and the other half of the offspring would look like the other parent. So then we can look at an answer choice like D, and if you remembered, oh, it's non-recombinant, then you would know, okay, that automatically means we'd see the same ratio as in the parents. So the second part of this answer choice wouldn't make sense to you. So that's another tip for taking test questions is if you remember a chunk of the answer or most of the answer and you're like, but I can't remember that last bit, sometimes, in the case of this question, that was really all you needed to remember. But I could have, made it a little more complicated. So try to remember both parts of the question, but also sort of logic your way through if you get stuck. You always want to go in knowing everything, but if you feel stressed, just take a minute and read it again. So now we're jumping into mutations and changes of the chromosome composition. So this one requires a little more thought. I'll give you a little more time to think, but try breaking down the answer options for this one and asking yourself what happens in each of these cases. What happens in a duplication? What happens if a cell is heterozygous versus homozygous for it? Ask yourself these questions as you're trying to figure it out. Okay, so what did you guys come up with for this one? So I heard E. So only A and C, which would mean it occurs during a heterozygous duplication and a heterozygous deletion. So why is it not B? So it occurs during a heterozygous inversion, but not a homozygous inversion. So think about what it looks like when it loops. And this is one of those points when I said, make sure you read the answer choices. Because you might be tempted to just want to look straight at D if you let this trip you up. So it occurs during a heterozygous inversion, but not during a homozygous inversion. What usually happens when you have a homozygous inversion, by the way? So again, this asks a question of lethality. So make sure you keep in your mind which of these is lethal, because a lot of times if you're like, I can't remember if the heterozygote has a loop or not, but if you remembered that, oh wait, no, I think one of these answer choices is lethal, then that might answer your question for you.
Are all homozygous duplication inversions lethal?
That's a great question. Can anyone pull that up in their notes? Are all homozygous, like duplication, inversion, deletions, are all of them lethal or just certain ones? So I heard no. Which ones are lethal and which ones aren't?
Deletion is lethal, inversion, I don't think duplication is.
Okay. So that's something to review, and make sure to go by what your textbook says and what your lecture says on this, that which ones are lethal. That's really big in genetics, which ones we see and which ones we will never observe because it kills them before they come to term. So, pretty important. So this is one of the ways to ask questions that sometimes confuses students, so make sure you read the question real carefully. Okay, what did you guys come up with? So keep in mind this question asks for what is false. Which of these is incorrect? So which of these did you come up with for incorrect? So I heard B. So why is B wrong? So I heard up here that it decreases transcription levels. So it's a regulation mechanism in eukaryotes. And we see over here that in what cells is it used as an immune defense process? In what type of cells? So in bacteria, it's used as a defense mechanism. Typically, their genes are just all methylated versus just some of them. What processes in human cells requires enzymes? Everything. So if you see something that's like, oh, this doesn't require enzymes, everything sort of requires enzymes. So that was, again, sort of something from Bio 1. So your body would basically come to a standstill if none of your enzymes worked. And then this is more towards later in the in your content, but this is cumulative. The idea that in eukaryotes we can use methylation patterns to inherit your epigenome. So you can sort of pass it along. Quick question from chapter 25, sort of the oddball out from the rest of what we're doing. My apologies. Yeah, feel free to speak up if ever I'm like writing off the screen or something's cut off. I can't really zoom in much farther. Also, if you can't hear me, again, just like wave. Okay, so for this question, we had this small island population of skunks, and a drought comes along, and it starts cracking down on all their resources, and as a result, their population dwindles to a very small number. So what is this an example of? So what is the bottleneck effect? So by random chance. So was this random chance? If an inside influence comes? Or an outside influence? Okay. What's migration? So they're leaving. What's genetic drift? I'm sorry, I can't really hear you. You have to speak up a little bit.
These already small populations has something happening in order to die that alters their gene frequencies in the population.
So, okay. And then mutation, sort of self-explanatory. This was the sort of weird one. And then what about natural selection? What's natural selection, going back to your Darwinian theory? So I heard evolution. So this survival of the fittest is the common phrase. So you guys said bottleneck. So where something happens and suddenly we shrink our population down. So I will tell you that one of my favorite types of questions to ask are enzyme functions. I've tried to ask them through the semester. So there's going to be a couple on those coming up. So does anyone remember the other name for topoisomerase? So gyrase, everyone remembers this one. Great. So that's the other name for it. So what's the function of topoisomerase or gyrase? So D, coiling and uncoiling the DNA double helix. What's one of the processes we learned about where we see gyrase? So I heard replication. So speaking of replication, what's the enzyme that does C? So helicase. So it's got the word helix in its name, and it unzips the DNA double helix. So make sure you can recognize it by both names. Your professor is totally, fine with putting either of these. I mean, I don't know if she will, or he will in the case of Dr. Stero, but could happen. So which of these is correct about facultative chromatin? So which one? B. So again, break down the two parts of the answer. It's not always present. So something doesn't stay facultative chromatin. And that one of those examples is X inactivation. So think of facultative, it's sort of like the word facilitate. So it happens to sort of assist or to allow for something to happen. So facultative, it's not always there, but it can happen over time, like X inactivation. It allows for X inactivation to happen. What's C? What kind of chromatin is that? That's permanent and makes up our centromere and telomeres. I heard constitutive. So constitutive sort of sounds like things like constitution. So it has to do with what it's made up of. So it's a little different from facultative, which can help, can facilitate. Constitutive, it's sort of like the building blocks, like our centromere, it's built from that. And then our telomeres. Do we transcribe the genes, or are there genes to transcribe along our centromere and telomere? So, no. So remember your different facts about the different types of chromatin. So this is one not so much on enzymes, but maybe about proteins. So give this one a whirl. So for our telomeres, they have this uneven overhang. And what is the name of the protein that protects those from the cell naturally wanting to repair those uneven overhangs? So C, shelterin. What does Shigoshin do? Because I'm guessing if you didn't put shelterin, you put Shigoshin. What does Shigoshin do? So I heard something about the centromeres. So what does it do for our centromeres? So keeps them together. What would happen if they weren't together? So B flip. So it protects them. So Shugoshin, Shugo in Japanese means guardian or protector, and Shin means god, so it's named after sort of a god of protection. And this protects your centromeres. Whereas shelterin will shelter your telomere ends. And then A and E, those are just nonsense as far as we know. I just sort of came up with those because they sound similar to shelterin and Shigoshin. What about Sen-H3? So has to do with histone assembly. So watch out with Shigoshin versus shelterin. So now we're moving on to replication, one of our three big processes. So for this one, I ask you, which of these statements is correct about DNA replication? So which of these is correct? So say it a little louder. So A. So only the new strands on the leading strand are synthesized in the same direction as the movement of the replication fork. What about the new strands on the lagging strand? So then the lagging strand is in the opposite direction. Why is D wrong? So yeah, they're both done in 5:3. So 5:3 is that big overarching thing. When we synthesize new strands, we do it 5:3. So I mentioned I like enzymes, so here's an enzyme question. So which of the enzymes that we talk about in replication, still heat replication, is able to proofread what it's doing? So which one does that? So I heard A, B. So which is it? B. So watch it between the difference between 3 and 1. So remember that some enzymes have multiple functions. They can do multiple things, like you see topoisomerases come up when we're talking specifically about chromosome condensation and decondensation and coiling. And it also comes up in replication. So we call it gyrase then. So keep that in mind. Some of these enzymes do multiple things and appear multiple times. So this is on transcription. And again, read these carefully. This is a bit of a detail question. I will say if you've attended any of my reviews before or used my materials, you might recognize maybe like 2 or 3 questions out of all these. So we're looking at the transcription apparatus for most bacteria. Which statement describes it? So C. It's 5 polypeptides. With the sigma factor, you may see it as this kind of like funky looking O with a little stem coming off of it like a cherry or an orange. That's sigma. And then it makes a hollow enzyme. So you can also think of it as the functional enzyme that requires that sigma factor. So another question on transcription, but this one's about termination. Question? Okay. It does say beta factor. Oh, theta? Beta? Not necessarily anything. Some of these questions I may throw in like words that aren't actually something, just to see if you remember which factor, so you don't just memorize it as, oh, a factor comes in. Remember, for some of these questions, your professor will say you don't need to know the name, and other ones she will mention the name. So keep that in mind. Theta comes up, but that's more towards replication in bacteria. So, good question. So, for row-dependent termination, so another one where you need to take a second and read it. Which of these is incorrect? This is again another one where I sit with the false. Oh, do you guys need another like 20 seconds? So, I heard a lot of people saying D. So, I heard the O's. Yeah, I heard from some of you that Dr. Bayer likes to put sort of the which one's false, which one's incorrect, so I tried and incorporated in a lot of those. Yes, make sure you read the question. And also, if you were able to see, maybe the question didn't make sense, if you were just like, wait a second, multiple of these are correct, then maybe you want to reread the question and say, Oh, it said incorrect. Okay, that's why. So when do we see a hairpin in termination? So row independent. I'd like to remind Dr. Bayerston specifically that some of her slides do have errors that she pointed out in lecture. Make sure you look back and reference your notes, or if you missed a day or something, ask your friend. Because I believe one of the errors is in row independent. But remember, she made a distinction. So if you're pulling up the slides and you see something might be off, that's one of the errors she pointed out from the diagrams in your book. Refer to the actual text of the slide and refer to the text in your book for that. So please keep in mind, the same with Dr. Sotero's, just as a general reminder, keep in mind some of the figures are not your professors, they're the books. If your professor said an error, make a note of them. Okay. So what is the function of the RAT1 protein? So C operates in eukaryotic termination. So here there's two parts of it again that has to do with eukaryotes, has to do with termination. What does it do specifically when it's part of termination? So chews up the mRNA. So I've made this sort of analogy before, but it's sort of like a rat. So it comes along and it eats up the end of the mRNA to an extent. So in eukaryotes. So what is the function of the 5' cap? So why is it C? Why is it not A? tRNA. So do tRNAs and rRNAs have 5' caps? No. So it's not in tRNAs. Does it help stabilize mRNAs? So, and then another part of it's helps with the recognition by the ribosome. So that's why it wouldn't be A and C, and it wouldn't be all the above. And we said it doesn't happen in tRNAs. Do rRNAs have them? No. So it's not B. So that leaves only C. Another enzyme question, this time in transcription. So which of these enzymes functions for eukaryotic transcription. So B, RNA poly 2. So this is one of those ones that was like a specific detail I know in Dr. Bayer. I don't know if Dr. Sotero covered it. So I would look back over your lecture slide if you said you didn't have to know that. There's that, but Dr. Bayer, she did mention it. So in class, she referred to a table and said you only need to know a part of it. There's a lot of detail on the slides, but you only need to know a chunk of the details, so refer to your notes. So this one's a detail question, sort of you knew it or you didn't. There's really not a lot of logic to this question. So what was it? A. So it was CCA. Why can you rule out C and E? Going back to sort of the basic differences between DNA and RNA. So thymine. So RNA doesn't typically have thymine. We have uracil. But again, it didn't even involve uracil or thymine. CCA, it's just something to memorize. And what attaches at CCA? What attaches at 3' N? So the amino acid attachment site. Okay, so we talked about a couple different types of RNA molecules. What is the function specifically of our small nucleolar RNAs, not nuclear, nucleolar, or our SNORNAs, colloquially? So which did you say? So B, helps in the assembly of our ribosomes. What does the splicing of introns? So I heard spliceosomes, and what are spliceosomes made out of? SNRPs. So small nuclear RNPs. So this is where you got to pay attention to the difference between SNORNAs and SNRPs. They're like 2 letters off. Yep. Okay. This is a question about the genetic code. So this is a question about our genetic code, things like your codon table. Why do we consider our genetic code to be redundant? So I heard E. So the amino acids may be represented by multiple codons. So if you were to have a codon table in the test, for this type of question, you could look at the codon table and see, okay, I see that there's a couple different codons that code for one amino acid. So a follow-up question to this. What is the effect that causes this? So the wobble effect. So we look at the codon and that sort of weakened pairing rule. So referred to as the wobble effect. And you can refer to your slides in your book for that. So this is specifically a question about eukaryotes and translation. So where do we see the start codon, in what sequence? A, so the Kozak sequence. Is the Shine-Delgarno found in eukaryotes? No. So it's only in bacteria. So again, keeping in mind, because remember, there are like entire chapters devoted to the differences between eukaryotes and prokaryotes. So make sure you double check the question and the answer options. So here you could have told right away that it wasn't the Shine-Delgarno. What does the Shine-Delgarno do in bacteria, by the way? So ribosome binding. So works in translation. Do you have a question? Yep. So you may hear them as the promoter and enhancer. The initiator, that's another thing that's just nonsense. So. So then it's for translation, so it would have to be in your mRNA. So when do we worry about the promoter sequence? In replication, transcription, or translation? So in transcription, and then the enhancer sequence? Transcription. And so what does the enhancer do? So it promotes transcription. So we worry more about the promoter, enhancer, silencer. So remember which one goes to which process. So ask yourself, what does the promoter do? And if your immediate answer is it functions in transcription, then. And double check what gets transcribed, what gets removed in mRNA processing. This is actually a question I asked in my last final exam review, and it stumped a couple students for a little bit, so decided to ask it to you guys. So this one, choose like the best answer, the one that is the real common link. So for how the genes of a single operon are related, what did you guys come up with? So I heard a lot of C. So function. So think about like our LAC-Z. What do all the genes in the LAC-Z operon relate to? What do they relate to? So digesting lactose, the metabolism of lactose. So they're united by a function. So these genes code products that relate back to their function. What's another commonality that isn't in the answer choices? So these genes share a common what? So they also share a common promoter. Are operons found in eukaryotes? No. So what are they found in? So operons are for bacteria. Now everyone's favorite or least favorite question types of operons. I can't remember if Professor Sotero went over this, so I'm sorry if he didn't, but I know this was in Dr. Bayer's. She went over this, so.
Can we go back to the last question again?
Yep.
If this is a common promoter, then wouldn't it have like the same basal apparatus, like transcription apparatus?
So bacteria use a basal transcription apparatus, and would the transcription apparatus change by the genes that you're transcribing? That's what you got to ask yourself. What do we say the transcription apparatus looks like in bacteria? So those 5 polypeptides in the sigma, did we say that that changes based on what operon it's at? So it stays the same. So it's not so much that, oh, these 5 genes are in an operon, and that means that they all use the same transcription apparatus, because the grand majority of all of the operons will use the same one. It's not so much that it's unique to the operon. Does that make sense? So we talked about how the most common apparatus is that one with the sigma factor. So that it's not so much unique to the operon as it's just the common apparatus. So that's why I said like that question was a little weird, because it was like the best answer. So for an operon that encodes an important enzyme, it has this accompanying regulator gene, and it encodes an inactive repressor. What type of operon is this? So I heard C. Why is it negative? I heard because of the repressor. Why is it repressible and not inducible? Because the repressor is inactive. So does that mean, is transcription normally on or off? So it's repressible because transcription is on. So remember that what the words positive and negative mean, and then break it down into what is repressible and what is inducible. And if you keep those 2 parts straight in your head, and you can put them together, then these questions shouldn't really give you a lot of trouble. And I'm going to give you another one. So now I'm asking about an operon of a structural protein with a regulator gene that encodes an activator, but it requires an inducer to come along before it can start stimulating transcription. So what type of operon is this? So B. Why is it positive? Is it the activator? Why is it inducible? Because it needs the inducer, so it's inducible. It needs to be induced. So is transcription normally on or off? So off. It needs to be turned on. So now we're looking at the lac operon, and now we talked about how there's more than one system of regulation for this operon. So when we're talking specifically about catabolite repression, whether it's functional or not, what two molecules do we worry about mainly? So cyclic AMP and CAP. So here's our pair for catabolite repression specifically. So make sure you can understand the process. Yep. So that's a good point. Is glucose also important for like for just in total as well? Yeah, for the lactose. So, but that's a good point. So yeah, for this question, I was specifically just asking, what are the two main molecules that we look at? Which one's the catabolite and that function? But yeah, I can see how this question, you could answer another one. Yeah. So why is it a repression system? So catabolite repression, it's not called catabolite activation. Correct me if I'm wrong on this. But the system's just called repression. So, but yeah, if it functions one way, it's not going to repress it. If it functions the other way, it is going to repress it. So just review your slides on the actual mechanism and what it's called. So. Whoop. So this goes back to one of our enzymes and when we're dealing with our forms of regulation. So what is the function of histone acetyltransferases, or HATs? So B, this is another one of those where you have to look at the multiple points of the answer option. So it's adding acetyl groups. What would it be if it was removing acetyl groups? So the deacetylases, not transferases, but the deacetylases. So it adds them to the histone proteins. It decreases the positive charge, allowing for the activation of transcription. So does it condense or decondense? Decondense. So it loosens it up so we can get in there and do some transcription. So that positive charge is what helps it keep all nice and tightly wound. So if we decrease that positive charge by adding in some negative molecules, it'll start to loosen up. I like to mention, because we have about 23 minutes left, if you have to go to a session at 12:30 and you need to leave a couple minutes early, that's totally cool. Otherwise, stay if you can, because we'll go through the remaining questions, about 5 questions left, and then it'll be open for you guys to ask questions. And then I also have some feedback surveys for you guys to answer real quick before you leave. So just to let you know, since there is at least one session going on at 12:30 for study union. Okay, so what is the function of the enzyme dicer? So A. So what types of RNAs are we dealing with when we use dicer? So the micros, what's the other one? So the SI, so the miRNAs and cRNAs. So now we're going to look a little bit more at mutations. So this is another one of those detail questions. This has to do with what are called transversions. So what is a transversion? So when you substitute a purine with a pyrimidine or a pyrimidine with a purine, what is C? So what'd you say? So a transition. What is a palindromic sequence, by the way, since that's important somewhere else? So it's read the same way, what? So forwards and backwards. So a palindrome is like an also, it's in words. It's when it's read the same forwards and backwards, so it's like symmetric. A word one is actually the word race car, where it's spelt the same forwards and backwards. So that's what a palindrome is. It's read the same whether you read it forwards or backwards. So this again has to do with types of mutations. So we have a base substitution that changes a codon, and the original codon corresponded to the amino acid serine, and then after the mutation, that codon still corresponds to serine. What type of effect is this? So what is a silent substitution, silent mutation? So the same, I'm going to say AA for amino acid. What's a missense mutation? So changes the amino acid. What's a nonsense mutation? And then what is a neutral mutation? So it encodes it's a different amino acid, but the difference between missense and neutral is that the one that the neutral mutation encodes is similarly enough that somehow the protein will still function similarly, unlike like a missense where it just won't make a right functional protein. That's sort of the new one. You've probably heard about the others before in different bio classes, but at least to me, the neutral is sort of the weird one to remember. So now some stuff from your biotech chapter. Should be fresh in everyone's heads. So, during the process of gel electrophoresis, DNA fragments, so E, they separate by size, and they move due to an electrical current. Why is C wrong? So, moves to the positive end. So your DNA molecules have a slightly negative charge, so they will migrate towards the positive end. Why do we see the, what size strands do we see more towards the positive end? So small. Why? So they just move faster. So when you look at the gel, they just move faster. So eventually, like if you just let this thing run for like forever, they would eventually all come down. But just that it moves faster through the gel. This one requires a little bit of thought. So when we're looking at blue-white clonal selection, what cells turn blue? So I heard E, I heard C. Which is it and why? So someone to raise their hand? So what does X-gal have to do with it? So that's what E, so you said that having X-gal allows it to cleave and turn blue. What do you have to say? Okay. So, did you have another thing to add?
Yeah, it's likely positive because the lactate gene makes beta-glycosidase. Beta-glycosidase makes
Okay, so why is E wrong then? So it's added later. So what he said was the LAC-Z gene, when it's not interrupted, makes beta-galactosidase. Which cleaves X-gal. So what is X-gal similar to? So if it's beta-galactosidase, what do you think that cleaves normally? Galactose. So X-gal, X-galactose. So it's just like a fake galactose. So you need beta-galactosidase to cleave it, and that will cause it to turn blue. So it's LAC-Z positive. So if a cell can't make beta-galactosidase in A, is it positive or negative for LAC-Z? So negative. What happens if they don't have ampicillin resistance? The cell can't grow on the plate. And if they were transformed by the plasmids, would they be positive or negative? Okay. So, like the bacteria, like bacterial transformation happened. So it took up the plasmid, generally. Okay. All right, so just review like your bacterial transformation. And then this is the last question. Like I said, after you guys finish this question, we'll have a question and answer period. And then if you would just stay for 2 or 3 minutes, we won't go over the 12:30, just to answer the surveys, get some feedback on the study union session. Which of these biotechnological processes requires DDNTPs, or D-deoxynucleoside triphosphates? So the Sanger method sequencing. What's another word for the Sanger method? So dideoxy, which would tell you that they use dideoxynucleoside triphosphates. So, okay, so again, if you guys could just like stay a minute, but make sure to be quiet for people who have questions. So does anyone have questions? We have about 9 minutes. Okay, if not, I'm going to pass out some of these surveys. If you could take a minute just to fill them out and give them back to me, that'd be great. So just like take one and pass it.
Is that the same thing as the barcode?
Yeah, but these are on my paper. Yeah. If you guys don't mind. And for those of you who are watching the stream, that would conclude this. I'm just passing out some surveys. I'll pull those sheets away from the .cam, and so you can see a barcode if you want to take it from home. Oh, you can just leave it on the end of the front table. Yeah, if you guys want to leave it on the end of the front right table in front of that whiteboard as you're walking out, that'll be just fine. Sure. As long as it's before 12:30? Yep, it's 12:25.
So with this question, I was saying in the case of linkage or not crossing over, what happens if it was crossing over? That's why I get confused.
So that's a great question.